All right. Thank you all for joining me here. We are having a fixation versus vitrification discussion, sometimes called ASC on one side or cryopreservation on the other. There's a lot of terms here that will also be clarifying a bit, but these are the kind of two main procedures that are used to preserve someone with the idea of bringing them back in the future. It's been a hot topic in this space about which one is better, what are the differences between the two, including on this survey, which is why we wanted to, on this server, which is why we wanted to have a discussion with the experts, because sometimes we get a little bit outside of our depths when we're talking about these pretty technical topics. So a few notes. Max and I are going to be moderating this discussion, and then at the end, we'll get to audience questions. So if you have questions, post them in the chat. Depending on how many questions there are, I can't guarantee that we'll get to every question because, of course, we want to respect the time of the experts here. They have important work to get to, but feel free to post them. And then I think there's going to be another mod who's sending me and Max the best questions. So if we don't pick yours, don't get mad at me. This discussion...
Get mad at the other guy.
Exactly, the other guy who I'm not going to name. It's one of the other two, yeah. So this discussion is being recorded and it's going to be posted afterwards, including on the biostasis standard, which I highly recommend you check out if you haven't already. The biostasis standard is part of biostasis technologies. It includes a lot of great resources like articles and podcasts that Max Moore hosts. So this recording is going to be released there. And if you want to check it out, go to biostasis.substack.com. I think Daniel should be linking that in the chat right about now, hopefully. Otherwise, we'll be linking it later. So without further ado, let's get into it.
Before we, first thing I want to do is jump into some definitions and everything. But before we do that, Andy, Aschwin, do you guys want to give us just a brief introduction into your own work and your backgrounds and what makes you interested in this topic?
Okay, I can start off. I've been sort of involved in sort of the larger biostasis, mostly cryonics space for more than 20 years. I've worked in the field pretty much in all different capacities, but I would say the most important thing I do is lead a research company called Advanced Neurobiosciences. And as the name entails, we specialize sort of in the brain and especially preservation of the central nervous system through low temperatures. So most of my research experiments is in sort of, the viability angle for preservation. Although a little while ago, sort of we have actually dabbled in chemical preservation as well. Sort of interestingly enough, when we really started our lab, we once did a sort of chemopreservation, and sort of that went really well, but we never thought about it very much. And then later, this approach started sort of really trending, and so I am pretty familiar with both, but of course a lot more with the viability sort of angle.
Hey, yeah, thanks. I'm Andy. I work at Sparks Brain Preservation. I've been here for 2.5 years, formerly known as Oregon Cryonics, formerly known as Oregon Brain Preservation. Prior to that, I did MD PhD training at Mount Sinai, and then I did psychiatry residency and research track, where I worked in Brain Bank at Mount Sinai as well. And I started volunteering at the Brain Preservation Foundation in the mid-2010s. And that was kind of my first formal introduction to the kind of brain preservation space. Yeah, so that's kind of my background.
Cool. Thank you both for joining us.
Mm-hmm.
So I'd like to start the conversation by first laying some groundwork that everybody can agree on, or at least if there are disagreements, we can unearth them rather quickly. And we can help our audience to understand a little bit more about the science here and what we're really trying to do. So can you guys start perhaps by defining explaining what chemical fixation is versus traditional cryopreservation with vitrification, And then things like the connectome, ultrastructure, and terms like that that we may run into in the course of this conversation.
Okay, well, I could, start talking a little bit about sort of what you would call the viability sort of preservation approach, right? And then maybe sort of Andy can sort of take over and sort of explain his approach. But I mean, traditionally, cryonics has been associated with halting metabolism through low temperatures, right? Because what we're both trying to do is achieve complete metabolic arrest. And actually, that might be an interesting topic later on, to which degree these approaches can completely stop metabolic activity of any kind. But in sort of, let's call it traditional cryo, you basically go to really low cryogenic temperatures to arrest any kind of metabolism. If you would do that with any kind of cryoprotectant, you will get freezing. So we introduce a cryoprotectant, usually through perfusion, to get to these very low temperatures and then store, until the time resuscitation attempts sort of can be made.
Awesome. Yeah. Well, let me first try to address some of Max's terms, and then maybe I could do like my kind of an overview like Aschwin did.
Mm-hmm.
Is that cool? All right. So fixation actually can be used to refer to many different types of chemical changes. Could just be with alcohols actually, but usually in this space, people refer to it as aldehyde fixation. So that's the use of like formaldehyde, glutaraldehyde, acrolein, glyoxal, that kind of a molecule that has been found to fix or harden tissues. That's what the original use of fix was, basically to harden a tissue in pathology. Formaldehyde first came into use mainly in the 1890s, rapidly spread across the world in the use of neuroscience and pathology in general. That's fixation. Vitrification is, of course, a phase change where when a liquid becomes so viscous, the molecules will effectively stop moving. Typically caused by low temperature in this space, but can also be done chemically. Connectome, that term refers to if you have a brain and you want to map well, actually, there's both a macroscopic connectome and a microscopic connectome. In this field, we usually refer to the microscopic connectome. So that's the synaptic connectome, or the set of all of the synaptic connections between cells in the brain. So you imagine the main way people have done that historically is they do electron microscopy and they do serial sections, and then they can map how each synapse, they can find the synapses and then trace from the synapse to the originating cell both the axon and the dendrite, and then you can build up a map of which cells connect to which ones and kind of their 3D structure. I usually prefer the term biomolecule annotated connectome, because as soon as you start talking about the connectome, people will say, oh, actually the synapses are not enough. We need all these molecules too. And that's true. However, it's not that big of a deal because all the molecules are there anyway. But I usually use the term biomolecule annotated connectome just to get ahead of that critique. Ultrastructure is just another term for usually used to refer to the type of images or structure you can only see under the electron microscope. People also use it for expansion microscopy these days. But just like really, really low level structures that need a very advanced type of microscope to use. It wouldn't be just like a regular light microscope. So those, I don't know if that was helpful to go over some of those definitions.
That was.
I think it's very helpful.
I think it would be helpful if you briefly, because we'll get more into it later, but explain why do we care about things like connectome and ultrastructure? What does this mean? What are we trying to preserve?
Okay, awesome. Yeah. So okay. So I have a few prepared comments. So I hope you'll indulge me. So first, I want to say thanks to Becca, Max, Daniel, and Eric for organizing and doing the cryosphere, and thanks as well to Aschwin for chatting today. I also have a few disclaimers. First, my comments here are off the cuff, so please take them with a grain of salt, and I apologize in advance for any inaccuracies. I also know this is a very sensitive topic for many reasons. And I want to talk briefly about why that is. First, some people think that only one type of preservation would allow them to survive. So obviously everyone wants to, or many people want to survive. So they want all the resources to be poured into that method. So people have very strong opinions on it. Second, people have a lot of career capital or money invested in these different approaches. It's hard to switch that kind of thing. So it makes sense that people are going to be really invested and care a lot about this discussion. And the third thing I want to point out is that most people in the world who are not in this discussion or not in this space think that we're crazy or delusional. So I just want to keep that, like, of course we're aware, we know. It kind of related to that, you know, long-term suspended animation is what people often kind of think of in science fiction. And that's kind of like, you imagine you go to a hospital and you say, okay, I'm dying of cancer. Take me to the doctor who could preserve me, and then I'll come back to life in 15 years. And it's like all done. It's like known. It's like, you know, they publish the results in JAMA. It's like this works, insurance pays for it. That's what we would all prefer, right? Like nobody wants to have to do this kind of thing where we go to these companies that like have to deal with all this stuff. Like, no, we want it to just be long-term suspended animation. And then they discovered that this thing does not exist. So long-term suspended animation does not exist, unfortunately. So then some people say, okay, this whole field is stupid. What is everyone doing? And that's a lot of people. And other people, mostly us, would say, okay, even though this long-term suspended animation doesn't exist, we can still preserve the information, or at least attempt to preserve the information that we care about in the person's body and especially in their brain. Generally, what people care about is their long-term memories, their personality. And what our neuroscience textbooks tell us is that if you want to preserve that information, then what you need is to generally thought of to preserve the biomolecule annotated connectome across the entire brain. That's why the connectome comes into play. So that's the disclaimers and the preamble. And I could stop there, or I could go briefly into why I think that the fixation approach makes the most sense if you accept those premises.
I think we'll get into the question of whether the fixation approach makes the most sense, or the more traditional cryopreservation approach makes the more sense in the course of the conversation. But of course, if you feel that we didn't address some specific thing that you want to talk about, please bring it back up, especially towards the end, because I want to make sure we get everything in. But I will say, Andy, you are among friends in the sense that all of us here care deeply about this subject, don't think it's crazy. And I know I speak for Becca as well when I say, I think we want whichever approach is going to work, right? Not necessarily, at least for us, we're not making money off of one specific approach right now. So that is a useful benefit in a very open and honest conversation about the pros and cons of any and every approach, including these two or any one that may be discovered or researched in the future. But I do want to ask, sort of as, so you touched on this a little bit, which is that, you know, we would all hope to get the sci-fi world where, you know, you lay into some pod and, you know, 5 years later, the pod opens, you look identical, you're a little tired, but, you know, within a few minutes, you're none the worse for wear. We don't live in that world, certainly not yet. So we have to deal with the world that we do live in. And when you were talking about this, you shifted over to the importance of preserving information, right? The information in our brains, the information in our encoded one way or another, whether it's through the wiring diagram that is the connectome or through the ultrastructure, but encoded in some way in our brains. I would venture to say some people, when they hear purely the term information, they think of themselves as more than mere information, even though in some sense that might be accurate. So how are we, can both of you comment on how are we encoded, or what do we know about how we are encoded in the brain, whether it's memories or experiences, whatever we want to call that, consciousness, identity, who we are, any of these terms you guys want to use or not use, but how does the brain encode or hold or run who we are on a moment-to-moment basis or over time? And how does that relate to these questions of preservation methods and the terms we've been using.
Yeah. I also want to make some sort of introductory remarks, like sort of Andy. Sort of in a sense, Andy and I are the best people you could have invited for this, and the worst. The worst is because we actually kind of agree, and I think Andy will weigh on this later as well. I personally think under the right conditions, both approaches can work, and I've coined that phrase for this, and that's called biostasis pluralism. I actually think under the right conditions, both procedures will work. So let me sort of start off by that, and we can get to that later. I think where it becomes really interesting is under suboptimal conditions. What are the interesting approaches here? But we can get to that. The other thing about information preservation, it is often said that if you preserve viability of a cell, neural tissue, or even the whole brain, then it is in tilt you must be preserving, the structural information, right? So the viability approach, I would say, would already automatically entail that you have good structural preservation. Now, of course, I think in traditional cryo, there can be actually a lot more effort in showing that it is actually the case. As to the question exactly how information and identity is encoded, I typically tend to just defer sort of to the mainstream thinking about it, right? And the sort of the mainstream materialist neuro-anatomical sort of consensus. To me, it actually does not matter a whole lot because we try to preserve the brain just as well as possible. And if we look at electromicrographs or we do viability and we see we can do better, then we improve again. And so we just want to, because by definition, the better we preserve the brain in all its details, the closer we must be then to preserving what matters. So that's sort of my pragmatic approach to this. I mean, there's people who really agonize about how exactly is identity and memory encoded. And of course that is important, but my approach is more like the better we preserve, the more likely it is that we capture that essence.
I do want to say, Aschwin, one point to that is the better we preserve. So how it is encoded matters for what matters to preserve, right? So if the ultrastructure and the weights of each individual synapse matter a lot less than the actual connections themselves, then we can say, who even cares to turn on the electron microscope? Let's just preserve other stuff. So I think in that sense, it might matter which technique and which approach we use if one is clearly better than the other on one of those factors. But Andy, I know you were going to jump in.
Yeah, so obviously we don't know for sure how the brain encodes who we are on a moment-to-moment basis. I think in neuroscience, we can make some basic statements. One is that neurons communicate with one another via synapses, and it seems like that communication, the electrical and chemical process of synapses and neurons communicating with one another, and the other cells that play a role, like astrocytes and oligodendrocytes, that somehow when these things do make their activity happen, that somehow leads to the experience of consciousness. How that is, I actually don't know. It's just a strange thing, but somehow it must happen. Consciousness has a physical basis, and when our neurons communicate with one another, that's what leads to us having these moment-to-moment experiences. Yes, I think if you were to have a loss of the structure so that you could no longer see how the neurons communicate with one another, if you didn't have the molecules or the structure in the same way as the person had during life, then I imagine it would be difficult to tell what the memories were. It's not a very clear way of saying it, but I'm trying my best here.
Yeah, I think you can sort of link sort of this preliminary understanding of identity and memory to what are these preservation methods, right? Sort of how does chemical fixation preserve or maybe even perturb or damage that, and how does cryopreservation preserve, perturb, or damage that? And then we can sort of look at what might be the better approach. Because as I said, in the end, there is no final word on identity and memory, the physiology of it, but we may have a reasonably good idea what chemical fixation does. We have a reasonably good idea what cryoprotectant toxicity does, or freezing, and we have to bring sort of these concepts together and see if we're doing a good job in preserving what matters.
Yeah, so it sounds like both of you kind of lean in the direction of we don't fully know everything that we need to preserve, but we want to preserve as best we can, and we think that both methods could work, but clearly both of you have some kind of preference, right? You've decided to pursue different approaches, so there must be some criteria by which you're evaluating why one might be better than the other. A few terms we've heard: viability is something, Aschwin, that you've brought up. Connectome obviously comes up in both, but I think especially when we talk about fixation. So let's get a little bit more into what are the actual differences between these two approaches, and why have you leaned towards one versus the other?
Okay, well, I can just sort of start off. Let's say you take a brain slice. Let's take a hippocampal brain slice, because that is presumed, the hippocampus to be important in, identity and memory. You can cryopreserve that with a cryoprotectant, and then if you do that well, you can rewarm and then sort of restore function. And there are a variety of assays you can do to see if that function is restored. And then you can say, okay, we can take this neural tissue to cryogenic temperatures, take it back, and we know it's functional again. That is, I think, also how most people sort of common sense think, right? If you preserve something, has it been retained? Well, yes, because you can reverse the procedure, and sort of there it is again, functioning in the same way as it used to before, right? And then the chemical fixation approach is through cross-linking, but maybe sort of Andy can sort of comment on sort of why he thinks that sort of retains that information upon reversal or reconstruction or whatever.
Yeah, so I guess the differences between the approaches, well, okay, so aldehyde fixation uses aldehydes, but you could also use cryoprotectants after and then do long-term storage. So it's could be a combination method. So I like to think of it as pure cryopreservation without aldehydes versus some kind of aldehyde approach. So, in the ideal case, you can imagine both ideal case where there's maybe no ischemia and a non-ideal case. Ideal case, unfortunately, is not legal anywhere in the world. You're not allowed to start the procedure before the heart has stopped beating. But that is what is done in nearly all animal experiments on both methods, both aldehyde fixation and pure cryopreservation. In the ideal case, we could imagine what would be the best validated method for structural preservation. So Ken Hayworth came up with this method that said, okay, let's look at electron microscopy data from the brain, and let's see whether we can physically trace, starting from the synapse, both the axon and the dendrite of all of the synapses in that electron microscopy data set. And let's see whether different methods can actually preserve this synaptic connectivity in a volume electron microscopy data set. And this was the Brain Preservation Prize. And the aldehyde stabilized cryopreservation approach won that, both for small mammal and rodents in 2016 and large mammal and pigs in 2018. But it's actually not surprising, because aldehyde fixation is the method used for connectomic studies. All of the connectomic studies use aldehyde fixation. So it's not surprising that they were able to preserve the connectome using that method. But I guess in hindsight, it's not surprising, but I don't want to discount the importance of that advance and that prize being won, because people did not know that was actually going to happen beforehand. So that's the ideal case. It's actually not that relevant to us, because we can't start the procedure before the person is legally dead. So then we have the 9 ideal case. And here I think the aldehyde fixation is the best validated approach for structural preservation as well. My evidence here is the brain banking studies. So basically, when people donate their brain to science, they often want to study the structure of the brain. And in that case, the investigators also use aldehyde fixatives, generally formaldehyde. And I believe there's been numerous studies using aldehyde fixation, including using perfusion fixation. I think the best one so far was a study from 2023 of Ricardo and Sausti. And they showed that you could preserve the structure very well, including at the electron microscope level, if the preservation was started within 24 hours after death. They didn't show anything like the Brain Preservation Foundation prize, but it's still, I think, the best available evidence in the literature for this type of structural preservation.
So when you say, and just to clarify, Andy, when you say this was demonstrating preservation of certain aspects of the connectome, what you mean by preservation is we can then look at it under a microscope and we can see the same structure is still there as was there prior to this process taking place. Not what Aschwin is describing as viability, right? You can't run action potentials through these neurons again anymore after you've done this procedure, or am I wrong?
No, you're completely correct. That's correct.
Okay.
Yeah.
Now, the one thing I'll say is we don't know that actually the structures are what they were prior to preservation because we didn't image them, but they are what we expect them to look like.
So Andy made a really interesting observation, and I'm actually glad he did. I told you we're going to agree a lot. It's like one thing I've often tried to explain when people sort of praise the virtues of sort of chemopreservation. It's like, yeah, they won the brain preservation prize, but that's also in a way their control and experimental procedure is the same, right? Because the preliminary prep for electron microscopy is their procedure, right? So there is no way how it could come out other than it has. So that's, I think, why it's indeed a really relevant discussion to talk about what actually do we expect from both these procedures in reality. And I want to say one other thing about the brain preservation prize, because of course there was traditional cryo and there was chemopreservation, and then traditional cryo didn't look so hot. But there was actually a reason for that, not to say that it could look perfect, and that is these were images that were made without opening the blood-brain barrier. And as probably a lot of you know by now, is if you don't open the blood-brain barrier, then the only thing the cryoprotectant really does is remove so much water from the brain that it becomes extremely shrunken, and that's why these images of the Brain Preservation Foundation for traditional cryo look that bad. So like that is really that can be a lot better sort of right now, and sort of images have been sort of generated that are a lot better. So I just wanted to provide sort of that context, that sort of the cryo images that they were compared against are not even remotely representative of what sort of we can do in the lab. In fact, I sort of it's not really for me to announce, but sort of my understanding is sort of in the next couple of months there will be finally a very comprehensive paper from 21st century medicine that shows good ultrastructural preservation with traditional cryo, which also should have actually the first ultrastructural results of a patient of a cryonics organization.
That's great to hear. I've heard about this elusive paper
Yeah.
for so many years that if it's finally coming out, I'll believe it when I see it.
Yeah, I understand the skepticism completely, and that's why I've said this before, and I'm of course I'm not the author, but I have now very good evidence that it has been submitted and all that good stuff, whereas before it was sort of still making the rounds and people had to finalize it.
Okay.
So
That's good.
Stay tuned for that, I guess.
Yeah, that's awesome.
And what we're trying to what we're trying to determine here is which of these two procedures better preserves the ultrastructure. So we're not really arguing about which one preserves our ability to reverse engineer the wiring diagram, the connectome, right? We know, so either one of these two is pretty good at that, but we're arguing whether chemical fixation or traditional vitrification preserves the ultrastructure better. Is that what we're just discussing here?
Yeah, I mean, I would say, actually, I'm almost making the argument sort of for any side. Yeah, if your procedure is identical to the prep that you normally do for electron microscopy, then yeah, of course, you will get a very, very good outcome. In fact, when Robert McIntyre, now Aurelia, was at the Elcor conference some years ago, he said, okay, here's the control, here's the experimental. I mean, which is which? Of course, no one could tell, right? Because sort of the prep is the same. Of course, what they put on top of it is the cryopreservation part, and that is maybe a topic we may want to dive into a little later on, because is cryoprotection of chemically fixed brains necessary for long-term preservation? Some people would say yes, absolutely, because there are still some long-term sort of, chemical changes going on if you would only do chemical fixation. And other people say no, maybe chemical fixation is good enough at room temp, maybe at refrigerator temp, maybe a little lower.
Can I ask this question, sort of going back a little bit, but should we care about viability?
Yeah, I think there are two reasons for it. One is the sole reason why we even have ideas what structures correspond to identity and memory, of course, is from viability studies themselves. So even, let's say, the structuralists are dependent on knowledge is being generated in studies that do recovery, or having rats go through a maze, removing some part of the brain and then seeing if memory identity is restored. So viability, even if you think viability doesn't matter for preservation, viability studies in general, functional studies, that's the only way to, of course, know whether memory identity sort of is preserved and which parts of the brain encode it. The other thing I like about viability is that it allows you to to tweak in a very fine-grained manner sort of the optimization of preservation method, right? So I wrote an article about that sort of a long time ago, and it's named Securing Viability of the Brain in Cryonics. And what I tried to do in that article is sort of to show at what point under ideal conditions viability is lost. Now, I think one of the big sort of arguments sort of in our field is like, if you lose that viability at some point, let's say halfway during cryoprotectant perfusion, well, why bother with it at all? Yeah, I think I could see the argument for that. But the other argument is, if you can show, for example, that you can take an experimental animal all the way to 0 degrees C and take it back, and there's just complete functional recovery, that I think inspires confidence sort of in a very compelling way, right? So you don't have to say like, oh, look at this micrograph and these are the parts that matter. No, something really comes back and is able to function. That is, I think, usually the kind of evidence that people sort of expect, right? And if people say like, well, these cryoprotectants are sort of kind of toxic, and they say, okay, now we have a new cryoprotectant and now we can get things back, as opposed to like a couple of years ago when we could not do that. So incremental progress is possible with these sort of viability assays.
I want to.
And Aschwin, how are you defining viability? Sorry, Becca.
Yeah, okay. So of course, viability is not sort of one thing, right? It depends whether you mean viability in individual cells, right? In brain slices, you can have small pieces of organized brain tissue where you can teach something and then sort of they function in the same way. You can have recovery of EEGs, and then you can have the ultimate procedure would be true suspended animation, where you take a whole experimental animal, or a human for that matter in the future, through these procedures, and you can reverse it and restore full functionality. So viability is not sort of one certain thing. You can apply it to different levels of tissue organization, right? Cells, slices, the whole brain, the whole animal. And that's another thing, right? Because sometimes people say, well, you're not preserving viability even in traditional cryo. But then the question is, what actually do you mean when you say that? Because for brain slices, you might actually be able to do that. For organized electrical activity in the brain, maybe not. Maybe that gets compromised with today's technologies. And whole animal, well, as we all know, that is right now still a pipe dream. Although, of course, it's actually one of my projects, sort of the cryo-DAO whole body resuscitation project. But that's a topic for another time, I would say.
Yeah. Andy, do you agree with that definition of viability?
Of course, I mean, he's welcome to define viability however he likes to. I just think it's a little bit unclear what he means when, yeah, basically to me, it's like if you preserve viability, then that is long-term suspended animation. And if you, because that's the way they use it in organ transplants, for example. In order to have viable organ transplants of the heart, you need to do the recovery within minutes. And that's how they use the term viability there. So if you use brain viability, I would imagine that brain viability is like something you could just take out. You just take out the brain and transplant it into a different body or something like that. But if you, but I guess yes, you could define it as like only the cells being able to replicate or something like that. It just, to me, it just if we talk about viability, we need to talk about exactly what aspect of viability we're discussing.
I think maybe a better distinction is viability versus functional recovery or global recovery, because viability is sort of pretty sort of common phrase, sort of in like cell biology and biological research, and it generally pertains to specific assays you can do, like mitochondrial assays, ATP, potassium, sodium, that sort of, you know, certain parts of an organ function, and you can go all the way to like isolated organ function. But I think in this context, probably what most people are interested in is complete functional recovery, right? Or maybe at least of the whole brain.
Okay, I want to start moving us in the direction of the practical, because we've started talking, we've defined these terms, what's the difference between these. We talk about, you know, ideal scenarios.
I want to cut you. I want to have a real quick.
Okay.
I cannot resist, Becca. I have to do one little
Go
thing.
for it.
I'm gonna help. I'm gonna try to help you, Andy. And you can say no, I don't need your help. But I want to ask to clarify a little bit more about viability or functional recovery. Let's say we could do this. Let's say we could take a fruit fly and we could determine, okay, here are the 200 behaviors that we see, you know, its clones performing in all these different circumstances. Okay, cool. We have a bunch of clones of a fruit fly. We see its behavior under certain circumstances. Then we take one of them, we do one of these procedures, and we basically destroy the brain of that fruit fly itself. But we are able to map its connectome and its ultrastructure and all the other things down to such precision that we then run a neural net of exactly what we just mapped out in incredible detail on some sort of computer. And then we don't do anything else. We just run the neural net in some sort of simulation of the same conditions that its clones, when they were alive, were subject to. And we see that fruit fly, neural net, simulated fruit fly, do exactly the same thing as its clones did while they were alive. Okay, so that simulated fruit fly, that's just that brain. We didn't do nobody came in and started programming more algorithms. It's just running the bare bones neural net of that fruit fly's brain. Did that stuff in the simulator. Did we now preserve some sort of viability function? What does that say? Does that say anything? Is this interesting at all?
I think it's super interesting, and it's the exact type of study that we want to see, that we expect should be done after fixation. This should be possible, and to a certain extent, it has been done in not like all of the behavioral assays, but aspects of the electrophysiology and behavior can be recovered from brain tissue, and that's exactly the kind of study that we think should be possible and hope to see more of in the future. I don't think people would call it viability though.
No,
but
I mean,
it's sort of the upload scenario, right? That's and to me, that's kind of if some approach is saying, look, we don't have functional viability, we can't turn the brain back on, but we can do this other thing, and we can prove it,
Mhm.
does that is that kind of the end goal then of a lot of chemical fixation, or is it, and is that what the difference is? I'm trying to really suss out the differences here between the two approaches in terms of how they do or don't care about viability, or how they do or don't define it differently, or functional, or whatever.
I don't think it's the end goal in terms of like how people would want to get brought back to life necessarily, but I do think it's the end goal. It's a very good end goal in terms of like the ideal validation that the method is working. So where you not only actually map the connectome, but you show that if you do a simulation based on that connectome, that you get the same behavior and you can infer the same memories were present in the brain as you would expect to prior to the preservation.
Aschwin, would you agree with that?
Yeah, yes, absolutely. I mean, of course, in terms of the traditional viability approach, sort of that I, represent, of course, what you need to do is, of course, straightforward, right? Sort of reverse the procedure and see if it's retained. So it's, I think, with scanning and modeling it and doing a simulation that requires a lot more assumptions, right? And that's, of course, why chemical fixation is often also linked to substrate independent minds. It's like, okay, I can see sort of this is working, but does it capture everything we care about, especially maybe sort of with humans in terms of consciousness and self-awareness? And the other thing that matters, of course, is um, if you would scan literally at a very high magnification a chemically fixed brain or animal, then you would have to let loose an algorithm that removes these crosslinks, I think, to get these kind of results in the simulation, because it would require things like, ion pumping and ATP or whatever, because these structures are not going to take care of themselves, right? It's still biochemistry, sort of, even if you believe in the structural approach. I think that sort of can be done, but that is to me actually an interesting question for people who believe in chemopreservation, but also in substrate independent minds as revival. It's one thing to say this level of structural preservation is enough, but how exactly will those crosslinks be dealt with in silico?
And when you say substrate independent mind, you're referring to brain emulation, or sometimes called uploading, correct?
Yeah,
Yeah.
yeah, and I think this is actually really important point to make, right? So it's not necessarily the case that a person who believes in the viability traditional approach doesn't think that structural preservation, chemical preservation cannot work, but there are other things, right? I mean, historically, um, chemopreservation, chemical fixation has been linked to mind uploading and substrate independent minds. I think most of the people who represented this approach sort of some years ago when it started really trending, like, Kane Hayward and, Robert McIntyre at this point, really linked it almost intrinsically to mind uploading. But that's, of course, not necessarily entailed. In fact, Robert Freitas' book Cryostasis has a scenario for biological repair of chemically fixed brains. And that is maybe something we also may want to talk about, right? Because all these things are always clustered in these ways, like, oh, you believe in chemopreservation, you must be a mind uploader. You're a mind uploader, so you favor chemical preservation. But these things are not exclusively entailed by each other, right?
Yeah, Andy, I
Yeah, I
do
agree.
I
Good
do want
point.
to hear your take on all that about the kind of link between uploading and fixation. But before we get into that, also, could you define cross-linking? What is that? Kind of expand on what's happening in the mind when you have cross-links.
Yeah, so I mean, so you have the body's made of a bunch of cells. The cells have billions of molecules. Basically, a cross-link is when an aldehyde, well, actually, not just aldehyde, there's all sorts of cross-links, but specifically an aldehyde cross-link is when an aldehyde molecule diffuses into a cell and just happens to find two chemical moieties that it binds to one chemical moiety on one molecule, and then a different aldehyde binds on a different molecule, and then they get linked together with covalent bonds. So it's literally just a covalent bond that's connecting two molecules that are already present in the cells. That's what a crosslink is.
Okay, and that's happening just in fixation, right? That doesn't happen in the traditional cryopreservation.
No.
I don't know.
No. And of course that raises, I think, a really interesting thing, right? Because when you look at electron micrographs, you do not see the chemical cross-linking, right? So, but unless you use a very extreme chemical, you also don't see the cryoprotectant toxicity. So one thing that's actually lacking, that is actually very relevant to this discussion that we don't have, is a magnification that is so fine-grained that we would see the actual cross-linking, and we would see the mechanisms through which chemical cryoprotectant toxicity would affect biomolecules. And if we would look at that sort of graphically in images, then we might say like, that is a bigger change than I feel comfortable with, or like, these cross-links. Like, for example, some people say the process of cross-linking itself is not benign. It might either alter ultrastructure in a way or make something sort of not really inferable. That is actually not my position, but I've some critics of chemopreservation sort of has made that argument. At the very least, if you believe in biological repair revival, you have to break up billions and billions of chemical cross links to be able to do that. That's of course for sure.
Okay, I think this is the point where I'm going to start moving us into the practical. I think Max,
Yes.
you got your question.
I yield the floor.
All right, because of course we're talking pretty theoretical, also what happens in the lab and like ideal scenarios, and I want to kind of think about how do these things actually play out in the real world. And we'll start by asking, kind of building off what you just said, Aschwin, how do we evaluate how good a preservation was, both in practice today and what ideally would we need to be looking at to know if a preservation was high quality?
Yeah. Well, I think one thing that the traditional cryo people cannot get away with is saying like, oh, we have good viability, so we should just assume we have good structural preservation. So I think we are really grateful sort of to the let's say the chemopreservation people to insist upon better ultrastructural evidence, because if you have good viability, you must have good ultrastructure. At least that's the assumption. And in the traditional cryo community, I think a much better job can be done to produce that kind of evidence. Not just regular electron micrographs, but seeing if everything is sort of traceable in 3D. That is going to be, I think, very important for traditional cryo in the next coming years. And as I said, hopefully there will be a paper appearing soon that sort of sets the stage for how well traditional cryo can do that. And then there's the very completely, the very different question is like, under suboptimal conditions, and almost all preservation, whether it's chemopreservation or traditional cryo, will have to be done in suboptimal conditions, right? People go through a long terminal phase or cancer, out of hospital cardiac arrest. How do these different approaches actually fare under these type of real world conditions? And this is where I have put a lot of emphasis in my own discussion of these technologies, because as I said, I personally think under right circumstances both can preserve information sort of well enough to permit future revival.
Yeah, we'll absolutely get into that. But first, Andy, what do you see as the way that we evaluate a good preservation, both in practice today, fixation wise? Like, what's being done right now, and what do you wish could be done?
I guess take biopsies of the brain and do electron microscopy. But are you talking about a patient or in a research case?
In a patient.
Yeah, patient just evaluate the presence, the quality of the perfusion, and yeah, I mean surrogate outcome metrics, unless they consent to a biopsy. Yeah, I think ideally in a patient you would already have a good enough knowledge based on research cases to know how the aldehydes affect the brain tissue. But I don't think it's necessary to do a biopsy in every patient case. I personally don't. So I know some people do, but I don't think that's something that's necessary if people don't want to do that.
Yeah, maybe not in every
Once you
case,
get-
but sorry, a certain amount of cases you would want to do this, right? Because of course lab conditions are going to be different than real world cases.
Yeah.
Well,
Well,
not
one thing I mean,
necessarily. Lab conditions can be the same as real world cases, right? I mean, you just simulate the real world cases in the lab.
Sure, that's fair.
yeah. Well, that one thing I sort of want to mention is that brain biopsies. I think it's a really good tool, and it's my understanding now that Tomorrow Bio is starting to do that. Based on informed consent of their patients, and I think so it may have already happened. So there might be results sort of soon. One caveat about that, though, I mean, I've had some experience in electron microscopy of ideal and non-ideal conditions. In non-ideal conditions, you can always find a part of the brain where the electron microscopy sort of looks really good, right? So it needs to be looked at in conjunction with other stuff. And in the case of traditional cryo, you want to see a CT scan that shows complete penetration of the cryoprotectant sort of in all areas. If the chemo people also add cryopreservation, then you also want to see their CT scans and see if the cryoprotectant has penetrated all parts of the brain. Because again, you may always have a small part of the brain that looks reasonably good, but that's not always a good indicator that the whole brain is preserved in that matter.
Mm-hmm.
Yeah, so for evaluating perfusion quality, you can do a CT scan if you have some kind of contrast agent present in the perfusate. The other thing you can do just to evaluate the surface preservation is just to look at the brain, because if the perfusion works, then the surface blood vessels would be cleared of blood, and also it'll just have a different color. It's basically it turns pale rather than pink.
Yeah,
And you can see whether the whole surface of the brain is like that.
With cryoprotection, in one way we have it easy, or at least right now, sort of DMSO really works well, sort of as that sort of kind of contrast agent in CT scanning. So cryoprotectants that contain a whole quite a bunch of DMSO are very good for CT scanning to see, how well the brain is being perfused and what the concentrations of the cryoprotectant are. With chemical fixation, it might be sort of a little harder if you only do that sort of to provide evidence that the whole brain is adequately chemically fixed. That might be a good question sort of for Andy, sort of how to do that. But yeah, maybe a contrast agent there would work as well.
Yeah, so in research cases, we're adding a chemical called omnipaque that allows us to do a CT scan. It's iodine-based, basically.
Okay. I'm curious to understand from each of you, what is the delta between what you see as currently the ideal procedure in fixation and kind of traditional cryopreservation respectively, and how does that differ from what's actually happening in practice? So I know ASC, aldehyde stabilized cryopreservation, I think this is the one that won the prize, but I don't know that full ASC is being offered anywhere, at least the cryopreservation part. So I'd be curious to hear about that from you, Andy. And then Aschwin, from your side, I'm curious about, we've touched on the kind of blood-brain barrier opener and how that ideally should be used. So kind of explain what should be done in the perfect world and what's being done now.
Yeah, so just from my perspective, I think the real world, I just don't trust the quality of perfusion. And once there's some degree of ischemia, I'm just skeptical that the perfusion is going to reach the entire brain. Or if it does reach the entire brain, it might require dehydration to the extent that I don't know what the effect of that would be on brain structures. So that would just basically the problem is there's two problems with non-ideal cases. One is that perfusion is difficult, and two is that the cells themselves break down throughout the brain independently of perfusion. Now, it doesn't happen immediately, but it does happen.
So then if you're not perfusing in all real world cases, does that mean that you're doing kind of immersion diffusion fixation?
So we are perfusing, we just don't trust it.
Okay.
So we perfuse and we get the best perfusion we can, and then after perfusion, we do immersion. So basically, like whatever perfuse that you get into the brain, it's going to immerse from the inside out and the outside in.
Okay, and then
And ideally, you get perfusion across the entire brain, but if you don't, you're still going to get diffusion of the formaldehyde.
And then if you're not perfusion, that means that you're not going down to cryogenic temperatures with that fixed brain, correct?
So after you do, it's the same thing I do at tomorrow. Is after you do fixation, you can then do protected immersion.
Mm-hmm.
And so we offer two options at Sparks brain preservation. Actually, right now we're just doing refrigerator, but we're working on a protocol to go down to minus 20 degrees Celsius because we think that would be good for a century plus. Once the tissue has been fixed. But if people want liquid nitrogen storage, we also would do that. It's not necessarily recommended, but if people want that, then it's an option.
Okay. And Aschwin, could you answer that question? What's the ideal and the practical?
Yeah. Traditional cryopreservation, I think, has a disadvantage when it comes to delivery of the agent that matters, right? The cryoprotectant to the brain compared to chemical fixation, in the sense that cryoprotectants very, very strongly dehydrate the brain, right? Because of the existence of the blood-brain barrier. The blood-brain barrier is an amazing structure, actually, of tight junctions that helps us, keeping all the bad stuff out of our brain. But unfortunately, it also has very poor penetration of CPA. So I think there's no longer an excuse for sort of traditional cryo organizations not to very aggressively start using blood-brain barrier modification, because currently the only sort of structural evidence that we have is pretty bad at the Brain Preservation Foundation, what was submitted, because those were images of brains that were extremely shrunken. And some of you actually may have seen CT scans in traditional cryo that were done under good conditions with little ischemia, and you get to see these very shrunken brains. So that will need to be changed to sort of catch up with the quality of chemopreservation. And then the other thing I want to mention, Andy sort of articulated this so well, in the end, it all stands and falls by the quality of perfusion, at least for the ideal procedures. Both chemical fixatives and cryoprotectants are vulnerable to ischemia-induced perfusion impairment, right? So it's not like, one chemical has no problems getting to the brain and the other one does. So we're both struggling with. In my opinion, that's where traditional cryo may have an edge, and now it becomes sort of a little bit more nerdy. Is like cryoprotectants are very concentrated agents. Often we say they're both hypertonic and hyperosmolar, and because of that, they can recruit a lot of sort of water, back to the circulatory system in very ischemic patients. Chemical fixatives usually are not very highly concentrated, and they're either isotonic or maybe only a little bit more sort of hypertonic. And one of my concerns about chemical fixation under non-ideal conditions is you cannot properly wash out the blood, and then these chemical fixatives will either cross-link or at least make it really hard to keep perfusing in a meaningful way. In traditional cryo, you can keep perfusing, but the results are usually not really like as in the lab, where you get complete uniform and good penetration of the cryoprotectant. But combining long periods of ischemia with chemical fixation through perfusion is, to my knowledge and also my experience, a little often literally a mess.
I think it's much better validated, at least in the literature, for ischemic perfusion of fixatives than cryoprotectants.
Yeah, there is actually not a literature on cryopreservation of the ischemic brain, right? Maybe there are a couple of papers about cryopreservation of the brain. Doing cryopreservation under suboptimal conditions, ischemia, is actually one of the sort of specialties and topics that my lab has done. So we know a fair bit about it, and there are currently not good sort of resolutions to how to perfuse a brain well after prolonged periods of ischemia. The same thing as for chemical fixation, right? That's why at some point you just take that brain out and do immersion fixation, which, by the way, is, I think, a big argument in favor of brain only as opposed to neuro and sort of head only. Like an organization like Tomorrow Bio, if they deal with a very ischemic patient, they can take their brain out and submit it to immersion fixation. You could not do that with a head.
Mm-hmm.
Let me ask you both this. For someone right now who's looking at this field and trying to decide what they think about it and how to think about it, and isn't just looking at this from a purely scientific perspective or medical perspective, but is also interested in they themselves, right? So we know that Spark's brain preservation, et cetera, is gearing up to be able to offer something for more people. Who should be more interested in chemical fixation versus traditional cryopreservation with vitrification at this stage, given where the science is at, where the medicine is at, where the practice of this is at, where the organizations are at? Is it that one is always better than the other? Is it a price sensitive person? Is it somebody with a particular scientific or maybe philosophical worldview who should be more interested in one or the other? Feel free to tackle that. Whoever wants to take that one on.
Yeah, it seems like, and that's why I introduced that phrase, sort of biopreservation pluralism, biostasis pluralism. I do think there's a lot of overlap sort of between these two schools. It's sometimes, I mean, there are advocates of one approach that, let's say, don't talk so friendly sort of to the other side. But generally speaking, it's very easy, in my opinion. In fact, I will be co-authoring a white paper with Jessica Radley from Nectome about it. It's actually pretty easy to present a unified framework in which both approaches can be made to work and the same evidence can be brought to evaluating these things. So I think in the end, it depends a lot more on do you care a lot about things like standby? What do you think about future revival cost? Like, for example, in traditional cryo, like organizations like Tomorrow Bio or Alcor, they set a lot of money aside for future revival. In the chemopreservation space, that hasn't been much of a talking point. Some of them think, well, maybe these mind uploads are sort of so cheap that's sort of not sort of really important. But I would actually not so much focus on the preservation methods. I mean, as far as I understand the science, but more like how is this practically being implemented and how to think about organizational stability, and do you have any ideas about sort of revival? How would you deal with litigation and things like that?
Yeah, I think I agree with a lot of what Aschwin said. It's a very difficult question. I'm incredibly biased on it because I'm, an employee and likely to be on the board of advisors of Sparks. So obviously, I think, we're the best, but yeah, everyone's going to take that with a massive grain of salt. So it doesn't really matter what I say, I guess.
Well, make the case.
Yeah, I don't
tell
think
us
that.
why.
Yeah. Well, I mean, just narrowly on the question of fixation versus pure cryopreservation today, I will argue kind of a historiographical argument that I think people have historically preferred pure cryopreservation because of this push for suspended animation, and it is the only method that could lead to the long-term suspended animation. So therefore, researchers are more interested in it. That's where the research funding goes. People like Saul Kent or Greg Fahey, they want to develop a long-term suspended animation method for humans. There's a lot of money in it. And it's kind of like where the organizational inertia goes. But I think we need to think about what is practically possible today. What is the validated evidence in the literature for structural preservation? What can we actually accomplish now? And to me, the evidence is very clear that the fixation has a better structural preservation. Now it's possible that pure cryopreservation could also work. It's certainly possible. And if there were no fixation options available, I would definitely go for pure cryopreservation. To me, it seems more risky. without the validation.
I like my part, what I would say is this: I rather have chemical preservation if I would know there would be sort of no ischemia, than let's say a whole lot of ischemia and then traditional cryo. But conversely, if there would be a lot of ischemia, I wouldn't want chemical preservation. So the circumstances under which these procedures are being performed, to me, is a lot more important, because sort of the beautiful electromicrographs that you, let's say, see right now with structural preservation, I think you are going to see soon from traditional cryo, don't mean a whole lot sort of in the real world. So to me, it's really a matter of like, how are these organizations dealing with ischemia and standby? And that is, I think, I think, you know, he can contradict me if it's misunderstanding, but I think Andy agrees with me. It like, if you don't do any, if there are no efforts for really rapid intervention and stabilization, and even, you know, chemical preservation needs that to the same degree as cryonics, then you're simply not going to get these images sort of that you're promised. So that to me is the really the key talking point.
Yeah, I agree that ischemia, minimizing ischemia is incredibly important. Totally. Yeah, I could make more of a case for
Oh, sorry, go
our
ahead.
organization, but I don't want to take up the rest of the talk.
Yeah,
maybe a different conversation.
I do quickly want to jump in. What level of ischemia are we talking about here? So what do you, Aschwin, Andy, what level of ischemia do you think that we should be primarily focused on optimizing? Are we talking realistically, we're aiming for 2 hours, in the real world, 5 hours, 10 hours? What do you think?
Well, okay. So this is sort of a little bit of my subspecialty, right? So, I mean, we've researched sort of any kind of duration of ischemia at normothermic and cold temperatures and see what the results are on the ultrastructure itself, but also on the ability to perfuse a cryoprotectant, and also, of course, a chemical fixative. And what we found was that sort of up to an hour of normothermic ischemia, it's still achievable to have good penetration of the CPA. You go much beyond that, it becomes a lot more spotty, and then you get sort of, you see these CT scans where there's very non-uniform distribution of the cryoprotectant. Now, if you would have pre-mortem or immediate administration of things like heparin or sodium citrate, you can push that point sort of a lot longer. But I think your perspective on how much ischemia is tolerable to be compatible with good structural preservation is almost more of an important dividing line in the field, I would say, than chemo versus traditional cryo. And of course, we don't have anyone of Nectome here right now, but it's my understanding that Nectome is extraordinarily demanding when it comes to the conditions under which they do chemopreservation, right? I've heard thrown around like, if we cannot do it right away or would go beyond 12 minutes of delay, then we would not even perform the procedure. That's certainly not Andy's perspective, as I understand it. So and I think this raises a really interesting question, right? There are pretty big divisions within these two schools of thought, right? I mean, you can find much bigger divisions within chemopreservation camp and within the traditional cryo than you're probably having right now between me and Andy. Yeah.
Yeah, that's true.
Is one or the other procedure better depending on when we expect revival to be, or uploading, simulation, whatever, when we expect that to be available or possible versus not? You know, if we expect that we're all going to end up running on a computer somewhere, then does that change which of these two procedures we should be aiming for or interested in, or does that not change it in any way?
In my opinion, it doesn't, because you will have to use an algorithm to remove the crosslinks for a realistic simulation of what you just scanned. And in traditional cryo, you have to make some modifications to reverse maybe membrane damage or some of the biochemical effects of cryoprotectant toxicity but I cannot really think of a time scale where one capability would be available, let's say, to, fix those crosslinks in silico and not being able to deal with, cryoprotecting toxicity. Unless, of course, the damage gets very extensive, but that would then be the case for both approaches. So if you see in silico reconstruction and revival as a plausible revival technology, then I think you need to do fundamentally the same thing in both cases, and that is looking at structure and saying, okay, what was it supposed to look like?
I agree with Aschwin that there's no practical difference dependent upon what revival method you want. I will push back slightly. I don't think you actually necessarily need to image individual molecules or algorithmically reverse the crosslinks with the cross-linking method. It depends on, this is kind of, again, pretty technical, but if we think about the whole brain emulation, it depends on what level of scale separation we have, which is a technical term. Basically, like what level of detail do we actually have to image and model in order to produce a whole brain emulation that accurately reproduces the same function as the original person? And it may actually be at such a level that if we could just look at the molecule with an antibody stain, which we can do today, it doesn't require any algorithms, then that would be sufficient. I hope that makes sense.
Yeah.
Yeah, and do you think
That
that?
that is.
Go ahead.
Well, no, I just wanted to sort of conclude that thought. It's like, yeah, that is the point I've never really understood, and I mean, actually, like, I'm not sure I'm understanding that. It's like, yeah, of course, we can debate about sort of what level of magnification is sufficient for identity and memory. But if you want to do the real simulation, you need the actual sort of underlying sort of biochemistry or mechanics of these tissues to actually simulate it. So you would have to, remove those cross links. That it seems to me that way, but I could be wrong though.
Well, we all need ribosomes in our cells, but we may be able to replace those ribosomes with simulated versions that don't actually have any effect on the function of the emulation. So in that sense, like it doesn't really matter like exactly which component of the ribosome is exactly where in the brain. It may have the same. It may be something you can just ignore.
Yeah.
Yeah, this is
What I
the
mean
crux
more
of it,
is
right?
like,
What
yeah, what
matters
I
and what
mean
doesn't.
more is, yeah, what I mean more is not so much like, oh, sort of how essential are these various components, but sort of the way the brain works, like functions, right? In silico should mimic sort of how the way brain works sort of biologically, and then it has to function at a sort of much higher level of, magnification, one would think. But yeah, I don't know. This that's that's what I would be thinking.
All right, and then I have one more question kind of related to revival before we will get to questions very soon, dear audience. Don't worry, we're almost there. But and of course, anytime we're talking about revival, I understand you maybe can't speak with the most confidence. This is just something I want your off the cuff opinion on. We're all subject to be wrong, but do you think that the bigger difference in maybe revival time or how much of you that we really care about can be revived is not so much chemo versus traditional cryonics versus how much ischemia is there? So basically, in a very good case, so much of the right structures are preserved that we can just work on reversing the procedure and doing whatever it takes to do reanimate, versus in cases where you have a lot of ischemia, you need to do that, and you need to figure out how to reconstruct certain parts of the connectome or infer, where the memories are and fix that. So do you think that is where the big delta is? Or do you even agree that second part is possible? Or are you concerned that if you don't get it properly preserved in the initial run, that second group might just be kind of screwed?
Yeah, that's of course a very good way sort of, I think, to summarize a lot of these issues, right? Yeah, I think under ideal conditions, I think there should not be a difference sort of in when the technologies are adequate for revival. It's really the ischemia that throws in a wrench. And that raises the question for both approaches, right? Because chemical fixation cannot reverse prior ischemia, neither does traditional cryo. So then the question is, when are things still inferable? And I'm sort of relatively optimistic on that, but of course there are limits, right? If you throw someone in a volcano, I think that's instant information theoretic death. But between perfect preservation and throwing someone in a volcano, sort of where do you draw the line, right? And the first paper actually of my lab was characterizing the ultrastructural effect of normothermic and cold ischemia, and we threw out some numbers, so some very preliminary thoughts where we think information theoretic death would happen. Just ischemia, right? That is all nothing to do with preservation methods, and it's not minutes, it's not hours, but it might be several days. But at some point, these images, I don't even understand sort of what I'm looking at anymore, right? When you have minutes of hours of normothermic ischemia, you look at these images and you can see, oh, I know what is there, but it's damaged. I'm pretty optimistic that an algorithm, can do that reconstruction, but there is a point where that's no longer possible. And of course, we call that information theoretic death. Sometimes people ask, when exactly is that? Well, that's hard to say, right? Because we don't know exactly what future technologies are capable of. But what I can say is if we let loose an algorithm on damaged tissue and it cannot infer the original state from the damaged state right now, yeah, okay, that would be a little concerning.
I think I'll say ischemia. One thing I'll say on this is ischemia. We actually have some research on it, and Aschwin's done some tremendous research, and I was a part of a team that reviewed some of the research on it as well. But I think in general, we actually don't know that much about ischemia. There's still more research. I know I'm a researcher, say more research should be done, but I'll follow the trope.
How
Because,
convenient.
yeah, very, very convenient for me. But if you look at like Ken Hayworth's metric of can you trace the synaptic connectivity from one neuron to the next with a volume electron microscope, nobody has done that at different time points. And so that would be like an example of something where we don't know. There's a lot of unknown. The other thing I'll say is there seems to be a lot of variability, where some people have slower degradation of their brain cells or slower loss of perfusibility than others. That's what we found in our brain banking program here so far, where we've done many cases. And I don't know why that is, but I think it's kind of similar to the notion of biological age is not necessarily chronological age, and your biological ischemic time is not necessarily the same as your chronological ischemic time.
The circumstances under which people die, and that I think has been documented fairly well in the forensic literature, can really slow or accelerate sort of also rates of decomposition. So like, for example, patients with fulminating sepsis are often known to decompose really quickly. So that's sort of also things to keep in mind, right? As a biopreservation service provider, sort of what exactly is the reason these people died, and sort of how does that affect how we think about sort of, their ischemic progression, so to say.
Interesting.
Yeah. In related, this is a public service announcement. Brain death is probably the worst thing that can happen to somebody who wants to do biostasis. So if you have this notion that like, oh, I want to live longer, I don't want to like let the doctors pull the plug, that's like really bad idea.
So pull the plug. Yeah. Okay.
If you're brain dead, like immediately. I mean, I'm not going to make any medical recommendations, but just from a structural preservation perspective, that's probably the worst outcome.
All right, that is good to know. Well, on that
We need
public,
to start the pull the plug club here.
yeah, on that public service announcement, right before we enter audience questions, Max, do you have anything you want to touch on? Or Aschwin, Andy, is there anything that we haven't? Obviously, this conversation could go on for many hours more, but is there anything very important that you think that you would like to put out there that we haven't touched on yet?
No, except that I'll say that I have a friendly disagreement with Aschwin about the ischemic case. I think in the ischemic case, I personally would prefer fixation. I don't think that the fixative perfusion is actually going to like fix the blood clots in place so that you can't then continue perfusion. What we found is that fixation doesn't happen that quickly. Perfusion the fluid goes through the blood vessels very, very quickly, and fixation takes minutes to occur. So basically, like if you get a fluid into the blood vessel and it's pumping through, even though there's fixative in there, it won't like immediately stop the blood clot in place as soon as it touches the fixative. The pressure is sufficient to push the blood clot out, if that makes sense.
Mhm. Yeah, yeah, that could be true actually. I think my reason for favoring traditional cryo over chemical fixation in ischemic conditions is mainly because of the composition of cryoprotectants. Cryoprotectants are what you call uh, hyper. They have a very high osmolality. Like there are like 10, 20 times as concentrated sort of as the intracellular sort of fluids. And because of that, they can draw a lot of water out of cells. And one of the things that happens during ischemia is a lot of water accumulation that makes perfusion so hard. And you actually can do this experiment, actually. You can induce a certain amount of ischemia in an experimental animal and then perfuse a chemical fixative versus a very concentrated chemical. And actually, if you look at what morticians do and funeral directors, when they deal with a very edematous ischemic body, they use very, very concentrated salt solutions. Now, here's an interesting thing. So for Andy, in theory, you can make a chemical fixative that is very sort of hyperosmolar and very concentrated that sort of functions in that way. So it's not even necessarily mutually exclusive. It just happens to be the case right now that a chemical fixative is sort of a relatively, similar in terms of concentration, as the intracellular environment of a cell, and cryoprotectants are, 10, 20 times as concentrated. And I think that is sort of where they shine to some degree. If you have a lot of ischemia, both approaches cannot like really clear the brain really well and reach all parts of it. And that
Yeah,
actually raises a really interesting question. And Andy and I, sort of announcement, are collaborating actually on a project. If you have a very ischemic brain, what is smarter to do? Immerse that brain in a fixative, and then you could even do some cryoprotectant steps. That's optional, or do a straight freeze, right? Sort of compare these type of approaches. And I'm not completely sure on that one, and sort of we're gonna research and hopefully publish on that.
yeah, and we are adding mannitol to our fixative, and we're considering adding polyethylene glycol to for the specific reason that Aschwin pointed out. And Aschwin actually was the one who suggested the polyethylene glycol. So thanks for that.
Yeah, and I think cryo is a lot longer, right? So old school cryo. So I mean, what I think will happen at some point is that sort of the chemo people will start modifying their chemical preservation solutions in a way to deal with these kind of situations, right? Well, unless maybe you're like Nectome and say, well, we only take you when you don't have any ischemia at all. Okay, that's different. But my understanding is that's not Spark's brain preservation's attitude.
All right. Well, with that, I think we can transition over to some audience questions. I realize that we're almost on an hour and a half. So if you guys,
Yeah, I can go a bit over.
okay,
Yeah,
go a
I'm
bit
fine.
over.
I'm
Okay.
fine. Yeah.
And we don't have to spend too much time on these, but unless you guys feel like we should. But so we will power through many of these. Okay. So starting in no particular order from the questions that our helpful co-moderator has listed for us. And I'm going to use the names here. If anybody doesn't want that, they should probably tell the moderator now. All right. So Lyas asks, Andy mentioned that cross-linking reduces the glass transition temperature of brains. I wanted to ask by how much? I'm thinking if we aim for vitrification instead of as cool as possible, then if vitrification could happen at higher temperatures, maybe it would be easier to store.
Interesting question. I don't think that the glass transition temperature is dramatically affected by cross-linking. It might be like a small change, but however, I do think it could potentially be possible to do vitrification at higher temperatures, especially if someone figured out a way to do trehalose and got intracellular penetration. Then you could do vitrification at higher temperature. However, I don't think the vitrification is necessarily necessary, in my personal opinion.
Okay. Leaf asks, again, mainly for Andy, he says, are you aware of any chemopreservation studies similar to the cryonics C. elegans 1, which shows reanimation and long-term memory persistence? If there is none, when do you think one will exist?
Great question. Definitely not right now. If you go based on Max's question of is it possible to take a fixed brain, scan it, emulate it, and see that you still have the memory present or not, that there actually is a decent amount of people working on that in the neuroscience literature. So there's a offshoot of the Brain Preservation Foundation called the Aspirational Neuroscience Prize that is trying to basically like incentivize people to work on this exact thing. And I don't know when it will happen that somebody will be able to preserve a brain and show that you can extract a non-trivial memory from it. But I expect it probably will happen in the next few decades.
Okay. Laeus asks, Aschwin on blood-brain barrier. I understand that breaking them is necessary as of right now. I'm wondering how likely are smaller cryoprotectants to exist? Is being large required, or could we eventually find less toxic and smaller cryoprotectants that penetrate the blood-brain barrier better without and aren't and don't need us to dissolve it?
That's actually really an excellent question. And for a couple of years, A&B, Advanced Neurobioscience, my research company, actually had a project looking at cryoprotectants that actually do cross the blood-brain barrier. And that not only has to do with size, it just, some things have access to the blood-brain barrier and others have not. Unfortunately, what we found is that many of these cryoprotectants, so far that we found to be able to cross the blood-brain barrier without that shrinking, were a lot more toxic. Now that raises the interesting question, because in chemical fixation, that doesn't really matter. So you could actually use a chemical fixative and then introduce a cryoprotectant of that nature without the dehydration. So it's actually an interesting concept for the chemo people. But we're not done yet screening these type of agents. There are cryoprotectants that can cross the blood-brain barrier without shrinking, but so far we found most of them pretty toxic.
Okay. Anonymous asks, is it known you have to achieve a high concentration of M22 in the brain for vitrification? A high bar Alcor rarely meets judged by its own published CT scans. Is the bar for a successful perfusion using fixatives lower than a successful perfusion using M22, i.e., do you need less fixative to do the same job?
I expect so, but I don't have very good evidence to prove that. But theoretically, I agree with that.
When you cannot read certain parts of the brain, often you don't get anything there at all. So neither your fixatives nor your cryoprotectants sort of really get there, right? So it's not just a matter of concentration. Here's a really interesting thing, because we're talking about M22. Tomorrow Bio uses VM1, which is the other major cryoprotectant in cryonics, and even despite pretty long periods of ischemia, their CT scans show very, very good penetration of the cryoprotectant that I have not seen in the Alcor CT scans. And there might be a variety of reasons for that, but there may be something that Tomorrow Bio is doing right. It could be the cryoprotectant agent or something else that is not happening at Alcor that yields these better CT scans, because almost invariably they have better CT scans for their cryoprotected brains than Alcor does, at least at this point.
And if it's not the VM1 versus M22, what else do you think could explain that difference?
Surgical technique, how long you take to ramp up the cryoprotectant. Alcor so far has only CT scans for their neuro patients, and Tomorrow Bio doesn't do neuro. I mean, they do brain only, but they always go through the chest for surgery, which actually is an approach that I favor in general for both approaches. So these might be, yeah, those might be some of the explanations, but there's something really interesting going on here that I think has not been talked about a lot in the community, and that is, why do overall the Tomorrow Bio CT scans better than Alcor's?
Sorry, by the way, before we get to the next question, just I'm curious. Does Sparks go through the chest? What is the standard procedure there?
No. Our standard procedure is to cannulate the carotids and the vertebral arteries.
Okay.
Both, right?
Interesting.
Mm-hmm.
Yeah, okay, that's good.
Yeah.
Because often some cryonics organizations only sort of cannulate the carotids, and then it's either assumed or checked in a way that the vertebral, that there is an intact circle with, okay, it gets a little technical now, but I'm not sure, I would at least cannulate all 4 vessels. I mean, of course, if you go through the heart, by definition, you perfuse all 4 vessels. But I am personally not a big fan of the 2 carotid only approach, and that could be an explanation.
For pets, we do the aorta, but for humans, we found we get better results with the neuroseparation approach.
Okay. Anonymous asks, can you comment on the long-term safety factor of chemical preservation being that fixed brains are immune to thawing?
Yeah, so this is definitely a thing that some people care about, and they're concerned about the organization or some kind of civilizational collapse. I do think in that case, the fixed brain is more secure. However, the liquid nitrogen has stayed pretty stable. These companies have a lot of money. I actually don't personally think it's very likely that they're going to thaw again, but it's a possibility.
Yeah, I mean, this is one where I cannot deny that the chemical fixative approach is slightly more robust long term, right? Because if you don't have any liquid nitrogen, a chemically fixed brain is relatively stable. Maybe not over very extremely long periods of time, but a traditionally cryopreserved brain will very easily be subject to ischemia and decompose. So that's certainly an advantage of the approach that's objectively the case.
I think it's mostly relevant to like Robin Hanson style, like civilization is going to decline type of scenarios, which again, I don't think is super likely, but it's not impossible.
Yeah, and there is a question of even if the brain continues to exist, will revival still be possible in the scenarios where the world goes that bad in either case, right? Questionable.
Yeah, it would have to civilization would have to come back and be like, oh, there's this brains,
Yeah, and you would still need somebody, alive and well to look after the bodies or the brains in both cases, and to continue the mission. But yeah, it's possible for sure.
There's a really
you don't necessarily need someone to look after it. I mean,
there's actually really interesting question here. It's like some cryoprotectants in very high concentrations inhibit microbial activity, right? So there have been brains that have been preserved in glycerol for a long time, and they preserve sort of structure relatively well compared sort of maybe to other cryoprotectants. So that raises a really interesting question, and something we have started to research in our lab: which CPAs favor or disfavor decomposition more than others, sort of looking at this sort of scenario where it's no longer being vitrified. In which CPA are you safer than another?
I would say that even in the case of a fixed brain, you still need someone to at least maintain them in a warehouse, where so if you imagine some post-apocalyptic scenario where everything's coming apart and the rain is pouring into the place, I don't think the brains are going to survive one way or another. Though a piece of the building will fall and break them or something. So I'm assuming the brains might end up like adorning some mantle over a fireplace somewhere, for decorative purposes, and then somebody in the distant future will say, I think we can turn those back on, or simulate them, or emulate them, or whatever.
That actually raises an interesting question, right? Because I'm whole body myself, but I once wrote an article arguing in favor of brain only as opposed to neuro, because the argument has traditionally always been, I think, based on the Dora Kent case, like, oh, in neuro, it can more easily move, right? But I mean, who is going to take sort of a severed head in an emergency, right? Brain only, sort of, I can see that, a brain bank might say, okay, we store this and whatever. So when it comes to preservation of just only the central nervous system, I'm very much sort of a brain-only sort of guy, because at least I think I can imagine scenarios where these brains can be quickly moved and stored in a sort of socially sort of acceptable manner.
Yeah,
Although
head is tough.
I will push back.
Yeah,
Yeah,
head is.
head is really
Do you
tough.
mind storing the severed head of this organization? Yeah, sure. Yeah.
I will say, pushing back a little bit though, if you
Mhm.
have an entire body, humans typically see a body and they think person, they think something, and whereas they see a head and that's morbid, horrible. They see
Yeah.
a brain, I think they just think like piece of meat, organ. So I don't think they would give it the same kind of feelings of this is something that should matter to the same extent as like a whole person that's been properly preserved.
Yeah,
But
no,
that's a
absolutely. But that's
yeah.
why I'm whole body myself. I mean, whole body I think is the most recognizable. It's maybe not as portable, right?
Right, yeah.
And then brain, and then sort of neuro would be sort of at the bottom in terms of acceptable portability, so to say.
Pros and cons to everything
Yeah.
here.
Yeah.
Okay, so Lieus asks, Aschwin mentioned the lack of a bigger zoom than ultrastructure to really assess the damage done by cross-linking. What is Andy's position on this? He didn't mention it when asked. How could we improve quality testing of fixation?
Yeah, I mean, it kind of relates to what we were talking about with what level of detail is going to be necessary to capture the important information in the brain. And I think that it actually, if you're able to stain for individual molecules and their positions, which I think you would be able to do without any algorithmic inference of the cross-linking, then that would be sufficient if you just know where all the molecules are. I don't think you need to know molecular conformations, which I also think could be inferred after fixation, but would be a little bit more complicated. So I think that our methods for, I think obviously I would like if we could more easily image individual molecules, but I don't think that the fixation will be a problem for that.
Yeah, I think one thing I want to sort of point out is that we talk a lot about sort of preservation quality and what these approaches exactly do, sort of to like biomolecules and structure. And it's actually unfortunate that, and I mentioned it earlier, that where the action really happens, we don't show images of that, right, to people, because I mean, chemical fixation. I mean, I actually looked this up in preparation, sort of for today. We don't have images like live images, sort of that show that. We can only like draw out sort of molecules and sort of okay, here's my crosslink. Cryoprotectant toxicity, for example, pertains to like the perturbation of hydration shelf proteins. You can also not pick that up in electron micrographs. And although I do agree with Andy, it's really a handicap in discussing these both approaches that we cannot just show an image at that level where there is some structural alteration and say like, look, this is how it looks like in both approaches, because that is a magnification that is way, way higher than what we typically use to argue our case.
Okay. So P, the letter P, asks, cells aren't just bags of water with chemical concentrations. Are there fragile structures within them that could be responsible for storing some part of the mind? How well do both techniques preserve these? What are the chances of each method becoming mainstream given the cost? Which matters a lot if you want to save as many people as possible. Cryonics has been available for quite a long time, and it's very much not popular. It almost seems like two questions here, but you guys can take, I guess, first and second there.
Yeah. I mean, what really matters for identity and memory, as I said really in the beginning of this, I don't know, right? I can have sort of a guess. I do think when you really come down to sort of individual molecules and ions, especially if they're sort of not attached, I don't think that sort of matters a whole lot. I think one thing we can say, of course, there are medical procedures in which the blood and fluids are replaced by a different solution, and people sort of come back from that. And the fact that that happens tells you something, I think, what matters about sort of identity and memory. And that is, I think, a good reality check when people say like, oh, these very sort of exotic things sort of matter for identity and memory. And then you can always ask yourself, are there mainstream procedures or accidents or whatever where these things are perturbed or changed, and do people come back with full identity and memory? And then if the answer is yes, then they may not matter sort of all that much. What's the second thing that was sort of tied to this point?
What are the chances of each method becoming mainstream given the cost?
Maybe Andy can first address the first one, because they're so different questions, and then we go back to it.
Yeah, I agree. Cells are definitely not bags with chemical concentrations, but perhaps they are. I think the most important aspect of the chemicals in the molecules inside the cell is their location. So that's why they're not bags, because the location matters. But I think if you have the location information and the composition of all the biomolecules, then that actually is pretty much all the information you need. Are there fragile structures within cells that could be responsible? Yeah, definitely. I think ion channels, their locations, neurotransmitter receptors, that kind of thing, could definitely be a part of. I'm not sure if that's a part of the mind. I think that would just be kind of like a key component of the whole thing. And those would either have to be preserved directly or inferable from what is preserved. How well do both techniques? How well does fixation preserve the spatial location of molecules? Very well. It's the gold standard for preserving the spatial location of biomolecules. What are the chances of each method becoming mainstream given the cost? I can't speculate. I mean, I could. I can speculate. It's not like I can speculate, but I don't really care to speculate as to whether how likely it is to become mainstream, because it's so difficult to tell. I obviously do think cost is important. However, my personal experience over the past year and a half has taught me that cost is actually not as important as I thought it was. Because we started offering free brain preservation in, I believe, May or April of 2024. And actually, I had this thought that like everyone was going to come out of the woodwork and we're going to get like inundated with so many requests. And I honestly had that thought before we went live with this. And then it didn't happen. We have had very few signups for this free research program. So I think it is a component, and it's certainly important to me when I think about preserving my friends and family. I don't think I would be able to afford some of these very expensive things. So it matters, but it's obviously not the only factor.
Yeah. I think neither cost nor, and that sounds weird coming from someone involved in science, but scientific evidence are very big players when it comes to uptake, sort of mainstream. If you look at our demographic, you can very clearly see that we're sort of not normal, sort of in a variety of ways. I mean, you guys have actually done some of that research. I have sort of my own theory on sort of what prevents higher uptake of cryonics, and I actually think sort of the future alienation argument is like really important. If you just say to most normal people is like, would you want to survive if you lose everything you have, like your loved ones, your assets, your belonging, your future employment opportunities? A lot of people would say hell no. It's only this sort of unruly group of cryonicist or chemo people who say, oh yes, I would still do it, right? So for me, the resolution lies not in the technical or cost things, although these things, of course, are very important, but to convey a message that people can take it with them. In fact, you could even say cryopreservation or chemopreservation allows you to take, things with you, not make mistakes of the past, give you new opportunities. I think these are the kind of compelling arguments that we need to make.
Okay, we're running short on time, but we'll get just a couple more in here. Everett asks, is it possible to restore brain function, biologically revive, of a fixed brain? If not, doesn't that mean that the only revival pathway left for a fixed brain is emulation, copy? My understanding is all revival pathways that don't ensure the continuity of consciousness, or at least do so plausibly, or provide some very strong evidence that consciousness is actually substrate independent, which to my understanding is a major assumption, should not be considered an actual revival. most people wish to be revived instead of having a copy of themselves created in the future.
Yeah.
Oh,
Well,
I would say if you don't mind if I jump in, the the people who have proposed molecular nanotechnology, Eric Drexel, Robert Freitas, Ralph Merkel, they have all said or implied that it would be possible to do this on a fixed brain. So that would be a biological revival method on a fixed brain. Now, is that possible today? No. Is it going to be possible in the future? Maybe, I don't know. I'm not actually an expert in molecular nanotechnology, but it seems plausible to me based on what I've read.
yeah, I mean that is something hopefully could be a takeaway sort of from this exchange, right? Chemopreservation doesn't necessarily entail substrate independent minds or mind uploading, and traditional cryo doesn't necessarily entail biological repairs, right? So it can sort of both ways in both approaches. So yeah, you can do a biological repair on a chemically fixed brain, and in principle, I don't see any identity philosophical questions, especially if you take a very fine grade repair approach without a whole lot of replacement.
Great. So Martineau DB asks, Andy, do you currently add antioxidants to the brain preservation fluid, and or is oxidation a significant long-term concern?
That's a good question. The answer is no, we don't. I think at minus 20 degrees Celsius, it will not be a significant long-term concern. If we were to do temperature storage at room temperature, it might be something we look into more. But I'm always reticent to make changes to literature-based protocols. So basically, we're trying to use the methods that have been shown in the literature to work for decades across dozens of experiments, many studies. And if we're going to make a change, like adding something to that, I would be concerned that we would do something that would cause harm and lead to it being worse than the status quo. That's why we wouldn't just like decide, oh, this might help, let's add it.
Great. One more question, and I'm going to paraphrase this one a little bit. So the temperature in the room sometimes between the different camps here has sometimes been a little bit high. Given that there is a lot of overlap in our ultimate goals, why has this been the case, and what can we do to bring the temperature down, which is something we all really care about doing in this space?
I mean, the temperature has always been down between me and Andy. You know, I think we both think we're working on the same problem. I think we both, under ideal circumstances, think each other's approaches have a reasonably good chance of succeeding. But that's unfortunately not always the case in the field, right? I think there are some traditional cryo people who think chemical fixation is just like it's not going to work. What certainly is the case is that there are a number of people in the chemopreservation space who are very skeptical about traditional cryo to the point that saying that offering it is unethical. I think that's not particularly helping, because I do think we are occupying a very niche field where we're trying to do the same thing, and that is use metabolic arrest for your future revival. So just throwing good questions at each other is probably good enough than taking sort of a more hot approach.
Yeah, I agree. There's so much overlap. We should definitely try to collaborate and work together as much as possible. 100%.
Yeah, and I
Cool.
will
Like, for
point out
example,
that is why we wanted to have you two specifically, not just for your expertise, but I think the way that you're both able to have conversations is much needed.
yeah. Like an example earlier, when someone in the audience asked this question, like, how could you sort of tweak the solution that you introduce for chemical fixative? Are you adding antioxidants or whatever? Well, these are standard components for carrier solutions in cryoprotection. So we can talk to each other. So what makes sense? I have made recommendations to Andy. Andy has made recommendations to me. And I think like in many fields, there are extremists, but hopefully these will not be the predominant players, and it would be the more reasonable collaborators. As I
Well
said,
said.
I am a biostasis, pluralist. I personally really think that both approaches under reasonably good circumstances sort of will work. And a lot of it has to do sort of with temperament, your history, how exactly it will work in reality, sort of what you favor, and also what you like researching. I like viability research a lot, sort of myself. But if it would really turn out to be a dead end for one reason or another, then I could easily see myself switching perspectives.
Well, I will there were more questions, but I will mercifully end it there in terms of questions. You guys have been really good sports doing this, and it is greatly appreciated. Hopefully, our audience gets a chance to see and experience this, or the broader audience, not just everybody here, and that would be fantastic. Where can people find out more about the work that both of you are doing?
So in my case, well, Becca already mentioned it, right? So like I actually lead two organizations, Advanced Neurobiosciences, and mostly, of course, we would publish papers and I speak in public. Another thing I've been doing recently is the CryoDAO whole body resuscitation sort of project. So you can follow that to CryoDAO and subscribe to the Biostasis Standard Substack where we do a lot of our writing. So that these are a variety of ways to sort of keep track of sort of what I'm doing.
Yeah, thanks again for having me on and having us on. If you want to check out our website for Sparks Brain Preservation, it's at sparksbrain.org. We try to do a lot of research here, and if you go to our research page, so sparksbrain.org/research, I believe, you can see our research papers and check that out. That's kind of the main thing we're working on. I personally also have a Substack, so I gotta promote that. It's called Neurobiology Notes, and I talk about neuroscience, connectomics, psychiatry, other cool stuff.
And we'll make sure whenever this is published, wherever it's published, we can have those links down below in the description. Yeah, on behalf of everybody, thank you both. Thank you, Becca. Thank everybody for being in the audience and for your great questions. And we will now leave you both to go and do the important science. Please work on that hard because we all really want this to work one way or another. Thanks, guys.
All right. Yes. Well, thank you for having me. It was amazing. All right. Bye-bye.